Hi, grade 8. Are you fine? Today we are going to continue about the vaccine and development of vaccine. We study before the passive immunity and the immunization and the active immunity. This is the type of active immunity when we introduce to the body some attenuated a microbe, for example, and the body will construct and manufactures its immunity against this. This is that we call vaccine. Development of vaccine, common misconception that activation of the immune system result in a protective immunity. Multiple factors affect decisions when making vaccines. Um, as we see now for the vaccine against COVID-19, activation of specific branch of immune system, development of immunological memory. What is the role of memory cells in the vaccination? It depends on incubation period of pathogen, short incubation periods. Example for influenza, symptoms already underway by the time memory cells are activated, repeated immunizations with neutralizing antibodies. For long incubation periods, example for poliovirus, enough time to allow memory B cells to respond. Let we see now the immunological memory versus serum antibody level. This is the mean antibody titer, the amount, which means the amount of antibody as function of time, the variation of amount of antibody as function of time till we inject the vaccine. This is the serum antibody, the amount of antibody in the serum in blood, which increases in 1 month, then it decreases. For that, we should take more than two injections from one vaccine. Look at the memory. It develops about 3 or 4 months, and it produces an immunological memory which is durable, which lasts for the lifetime. Types of vaccine, all organism, for example, attenuated, viral, or bacterial. Attenuated means that we take out all the invading ability from the bacteria or virus, and it becomes attenuated. Or inactivated viral bacteria, it is similar to the attenuated one. Purified macromolecules, we take the pathogen and we purify the macromolecules from it, for example, the antigens. Polysaccharides, toxoid, which is the toxin, attenuated toxin, recombinant antigen, recombinant vector. Whole organism vaccine, such as for anthrax, for cholera, for many other pathogens. Many common vaccines used consist of inactivated or attenuated bacterial cells or viral particles. Includes attenuated and inactivated vaccines. What is the attenuated viral or bacterial vaccine? Attenuation, to reduce in force, value, amount, or degree, weaken. Achieved by growth under abnormal culture conditions. For example, Bacillus calmette-guerin, BGC, BCG, for example. act as double-edged swords as they have distinct advantages and disadvantages. Advantages of attenuated bacterial or viral vaccine. The advantage stem to their capacity for transient growth, a prolonged immune system exposure, single immunization, replication within host cells. For inactivated viral or bacterial vaccine, methods of inactivation include heat or chemical agents, and the result loss of replication ability, difficult to inactivate due to the potential for denaturation of epitope, dependence on higher order level of protein structure. Look at the attenuation versus inactivation. For the attenuation, normally require one dosage to induce relatively permanent immunity, primarily cell-mediated in nature, while inactivation requires multiple boosters, emphasis on activation of humoral immunity. However, something very important is missing. Thank you for watching. This is the end of our video, and see you in the next